because we need to go with the next presentation of Kamila Muchowska from University of Strasbourg. Hello, Kamila. Do you hear me? Do you see me?
Can you hear me okay?
Yes, it's perfect. Thank you.
Perfect, excellent.
So
I need to share my screen.
Yeah, Kamila Muchowska is our invited speaker and tell us something on non-enzymatic metabolic reactions and origins of life. The microphone.
I think you can see my screen now.
Yes, we see your screen, but not the presentation itself, but whole.
Okay.
Okay, now it's perfect.
You can see the audience view, not the presenter's view, I hope.
Yes, everything is fine. We can start. The microphone is yours, Kamila.
Okay, so good afternoon, everyone. I'm Kamila Muchowska, and I'm a CNRS research scientist at the Institute of Science and Supramolecular Engineering in Strasbourg. And first of all, I would like to thank you all, especially the organizers and the audience at the Life and Space conference, for inviting me to present the research that we carried out on the Origin of Life project at the Laboratory of Chemical Catalysis here in Strasbourg. Before we move to the scientific part, I would like to show you some highlights of where I'm working and where I spent the past almost 6 years. So Strasbourg is situated in the east of France in the historic region of Alsace, and the entire historic city center is now considered a UNESCO World Heritage Site. And especially people from the European Union might associate the city with the official seat of the European Parliament. And for those from outside the EU, this is one of the most important institutions in the European Union. And this is our institute, and we work here just behind this building. And now on to the science, but before I go to science, we need to go a little bit back in time by about 4.5 billion years to the point where life on Earth did not exist. So soon after the planetary accretion, I'm pretty sure the astro people at the conference will be able to tell you more about that. But what I can tell you is that after the planet accreted, the liquid water appeared on Earth. And according to most models, the atmosphere was full of CO2. The core of the planet was and still is molten metallic iron and nickel. But the further you went to the outside of the planet, the more oxidized the layers became. And this created some sort of a disequilibrium, which is quite important for today's story. So keep this fact in mind, and we will come back to it. And the general purpose of the research that I will present to you today is to gain a deep understanding of the chemical foundations of living cells. And to do that, we need to know how they came to be the way they are. My little disclaimer that I would like to make is that I will be talking about life as we know it, and the model environment will be based on what we know about the early Earth. So in other words, the question that our research is trying to answer is how geochemistry, available on the early Earth, self-organized to become biochemistry. So this is what you see in this circle, and how this biochemistry eventually became biology. So over the past decades, the majority of prebiotic chemists seemed to be of the opinion that in order to solve the origin of life problem, we need to find a way to make biomolecules from simple substrates. But in the recent years, it becomes more evident than ever that making life's building blocks alone is simply not enough. And this is because approaching the emergence of life as a matter of chemical self-organization, so this requires us to look at life as a dissipative process and not just a collection of static biomolecular building blocks. So what are these dissipative structures? These are open systems which operate out of thermodynamic equilibrium. Whenever we find these kind of self-organized systems in nature. These are, for example, a hurricane, a water vortex, or an oscillating chemical reaction. So they are always driven into existence by a continuous source of energy. And this continuous source of energy creates a gradient. So the characteristic dynamic structure that we observe, it forms because forming it is the kinetic channel by which the energy gradient can be released. And this is done through a path of least resistance used for that energy release. So in short, life is also a dissipative system like that, and it follows the same rules of organization as, for example, a hurricane. So hurricane is built from air and water, and in order to have this hurricane vortex structure, we need to apply a gradient across a 1,000-kilometer scale and the right temperature also. And as a result, we obtain a process called wind. So the same rule applies also to life's chemical processes. And we know the building blocks of life very well. But if we took all the necessary biomolecules and added them together, and even like stirred with a fork or a whisk or something, we will not obtain life. The point is that for life to happen, some disequilibrium in the environment was necessary for chemical reactions, such that they organize themselves into a process. And this process we know today as metabolic pathways. So in the living organism, metabolic processes are generally enabled by complex proteinic enzymes, like this one here in blue, the blue ribbon. And often these processes consume ATP. So you've got ATP here. So what I want to show you is an approach to the origin of life on Earth that we use. And basically, this approach is drawing a parallel between the general way that biochemistry operates in cells and how it might have operated very early on, when the enzymatic machinery or molecules like ATP, which are quite complicated, they just did not float around on the early Earth. So for this reason, instead of these enzymes that are big proteins, we will focus on much simpler catalysts available in the environment, such as, for example, metals or metal-containing minerals. Right, when I started to work on the Origin of Life project as a postdoc with Joseph Moran, our work focused on understanding where life's two oldest carbon fixation pathways came from. And these are the Wood-Jungel, and also called the acetyl-CoA pathway, and the reductive Krebs cycle, so that's the round part, which I will refer to as the RTCA cycle because it's just simply easier. So the linear pathway, I don't know if you can see my cursor. Laser pointer. Okay, great. So this Wood-Jungel pathway is the only linear one. And it's present in both bacteria and archaea, and it is the only thermodynamically downhill pathway. So it converts two molecules of CO2 into 2-carbon acetate, which is then used for other metabolic processes. And some people also include the 3-carbon pyruvate into this pathway. So this one has been hypothesized to be very ancient. And the reverse Krebs cycle, the RTCA cycle, for those of you who are not chemists, just ignore the structures and stick with me and I'll walk you through it. So this cycle is really important from the evolutionary point of view because it contains the 5 universal common precursors, such as acetate, which can give rise to lipids, pyruvate, which can be then taken on and metabolized into alanine and sugars, or oxaloacetate, which leads to aspartate, also other amino acids and pyrimidines, glutamate, which is extremely important in the metabolism of amino acids, and succinate, which then can serve as a building block for pyrroles and other cofactors. So it has been hypothesized that these two pathways were once fused and they worked together in the prebiotic world. So we wondered, can we obtain such a network or parts of it without enzymes? Because obviously enzymes as complex proteins, like I told you before, they did not exist on the early Earth. So on one hand, you might say that, okay, this is a bit unlikely to work because if we start from CO2 in water, CO2 is not very reactive, but you might also notice the green blobs I put here. These are the enzymes that perform these reactions in nature. So most of them use transition metals in the active sites, or they use transition metal cofactors. So transition metals are also used all the time as catalysts in organic chemistry, in organic synthesis. So the question is, for now, could we use metals alone to do the same job and promote these reactions? So that's the experimental setup we got. We organized some pressure reactors to be able to work with gaseous CO2, and we started. So first, we took the linear pathway to work with that, and we did some high-throughput screening with different metal catalysts to see if any of these reactions of the acetyl-CoA pathway, CO2, carbon dioxide, if it can be promoted or, we say, fixed into higher carbon products. And we were especially curious about iron because geochemically, like you saw, it's the most abundant metal and arguably the most important one for biochemistry. So it turns out that if we take metals that we find in the biological wood-jungle pathway, such as molybdenum, iron, nickel, cobalt, manganese, and tungsten, place them in a powder form in salty water and expose to carbon dioxide at 100 degrees, we can fix carbon dioxide into biomolecules, acetate from 2 molecules of CO2 and pyruvate from 3 molecules of CO2 in the case of iron, nickel, and cobalt. So the simple reaction directly follows the intermediates of the linear wood-jungle carbon fixation pathway that I showed you. And last year, it got even better, because together with our collaborators in Germany and Japan, we found that you don't really need metallic iron to promote the wood-jungle pathway without enzymes. It works perfectly well with the hydrogen gas and hydrothermal minerals used as catalysts. So you've got awaruite here, greigite, and magnetite, and these minerals can be found in hydrothermal vents at the bottom of the ocean. So this way, the potentially prebiotic carbon fixation pathway that runs without enzymes, just promoted by the minerals, this way it becomes really similar to biology, because in biology, hydrogen is also used in the analogous process. Right, so can primordial metabolism work without enzymes? The answer is yes, at least for this part network, linear one. So let's go on then. In our work on the RTCA cycle, that was a bit more complicated because there are more reactions. But we started with the irreversible reduction reactions, which you can see here. They are marked with the pink numbers. For non-chemists, ignore the structures, the reactivity, just focus on what I will going to walk you through. So we use the irreversible steps to couple them to the reversible ones. So these can go back and forth. So we use the irreversible ones to push everything in towards one direction to obtain sequences. So we split the cycle into two sequences. You've got the bottom sequence in black and here as well. And after screening thousands of reactions, I would say roughly over 4,000 for this project, what we found is that using metallic iron, zinc plus 2 salts promotes uniquely not just single steps, but entire 2 sequences. So basically, you start from this, you put the catalyst, you wait, you heat it, under acidic conditions, you end up with a mixture which contains this product. And the same thing with the upper arch. It works exactly in the same way. Metallic iron, zinc, and chromium salts under acidic conditions. So this shows that you can perform the whole sequences of biologically relevant reactions without enzymes and also without human intervention after every step. Now I want to show you something for non-chemists, what it looks like when I talk about medium to high throughput screening in this type of project. So we run dozens of reactions in those little vials and dozens at a time. We change different parameters like temperature or additives, and then we triangulate to find the optimal conditions. So most of these reactions were analyzed by gas chromatography coupled to mass spectrometry, or by NMR, which you can see here. So basically, you just run lots of reactions and narrow down to the best parameters. So to recap quickly on where we are. So after screening nearly 10,000 reactions for both of these pathways, we found that the combination of metallic iron and transition metal salts could promote all of the ATP-independent steps of both pathways. What I mean by that? In biology, some of the steps require ATP. The problem was that we were not able to promote any of those steps that are ATP dependent in biology. And most of these happen to be carbon-carbon bond formations. Well, building a carbon skeleton is necessary for anabolic processes, right, and in metabolism. So, so far, now that I know, no ATP-dependent CC bond forming reactions had been shown to have prebiotic analogs. And it's quite unclear whether this is because these reactions are extremely difficult without enzymes, just from a kinetic perspective. Or is it simply because such an experimental approach to prebiotic chemistry is, maybe it's fairly new and maybe not many people have tried to have a go. So then we wondered, how did EverLife come to form those carbon-carbon bonds? What I mean is, for example, here, when you have a 3-carbon molecule, and here you have 1, 2, 3, 4. So basically, you're adding carbons. And this is apparently quite difficult. Some time ago, quite recently, we proposed a possible solution to how CC bonds would have formed in biochemistry without the need for complex molecules like ATP. So this is quite complicated, but major message of this slide, the main message, is that simply mixing 2 compounds, pyruvate, which has 3 carbons, and glyoxylate, which has 2 carbons, in the presence of iron plus 2 salts, so basically iron chloride, in warm water, gives rise to a complex reaction network. So here you have, first I want to show you the level of complexity. So after 24 hours, you basically obtain all these compounds. And pretty much all of them are biologically relevant. What this graph shows is that basically the higher the complexity, the more compounds you get, the higher also the mass loss. You have decarboxylations, you lose a lot to CO2, it just bubbles out. So the breakdown processes also begin to be very visible. The correlation between the growth of complexity and the loss of mass is there. But the fact that you have so much breakdown in this experiment is actually not a bad thing. And I will explain this in a moment. But first, for those of you who are into chemistry, let's have a look at what this network looks like. So you start from here. Here, glyoxylate, 2 carbons, pyruvate, 3 carbons. After 24 hours, without any further human intervention, you just wait. The network grows, and then it starts to perform these side reactions, and these, and these. And some of these are decarboxylation, so obviously it collapses a bit, mostly to CO2. There are also other smaller products, but this is not relevant at the moment. So finally, if we introduce a source of nitrogen in the form of hydroxylamine, we can also obtain 4 biological amino acids, which are obtained directly from the network. And these are alanine, glycine, glutamic acid, and aspartic acid. To give you a better understanding of what I'm talking about, why this network is so relevant, here is the same thing as on the previous slide, but redrawn and overlaid over 2 biological carbon metabolism cycles, the Krebs cycle and also another existing biological pathway, the glyoxylate cycle. The fat arrows highlight the reactions present in our network. So here the red fat arrows, 9 out of 11 intermediates of the Krebs cycle are covered by our network, 7 out of 11 reactions, and also 8 out of 9 intermediates of the glyoxylate cycle, and 5 out of 8 reactions. So we are really starting to sort of recreate biochemistry without enzymes, and it's one that operates on an inorganic promoter instead. So now I said that before, it's actually not so bad that the intermediates of the network are being broken down. And this is because life builds up molecules and breaks them down in a steady state. Just like this fountain in Strasbourg pumps water up and down and lets it fall down under gravity. So the goal of our study in principle was to demonstrate that an abiotic system could show just that. Molecules made from CO2 can react to build up complexity, and at the same time as they break down again to smaller products, they are converted to CO2. So
Kamila, you have 3 minutes.
I'm almost done. That's almost it. So yeah. And this brings me almost to the end, as we know. So of course, this is simplified biochemistry, largely based on carbon metabolism, and it's only a small part of the metabolic map. And this metabolic map only describes what we know about life as we know it. But I think it's a great starting point for exploring a bit further. So now what the Moran lab is working on is going beyond the RTCA cycle. So towards here, 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 and here. And I personally am more occupied by looking into the beginnings of biocatalysis. One thing that comes to mind that I would like to still mention as a general remark is a phenomenon that, to me, it reminds me of a biological version of the butterfly effect. And what it means is that throughout the emergence and evolution of biochemistry, changing a tiny thing in one place, so for example here, could lead to really catastrophic changes somewhere else, to something that is kinetically coupled. So for example here. So these networks are a bit like this as well. So this is a reason why I think the core of the current biochemistry, so for example this part, has stayed practically unchanged for billions of years. And thanks to this, we can look at the most fundamental biochemical processes, and we can extract some hints about how they evolved. To summarize, the main conclusion of this talk, regardless of the fact if you're a chemist or an astroscientist or a biologist, is that experiments show that metals can promote non-enzymatic analogs of core biochemical pathways. And of course, there's still a great number of fundamental questions about the nature of life, and we're just beginning to scratch the surface. But while we do this, it's important to remember that, I really like this quote, to understand the origin of life, it would be foolish to ignore life as a guide. So let's remember about that, and the work ahead of us will definitely be really exciting. So with this, I would like to thank everybody who has been involved in these projects over the years. And this is a quite a bunch of people, especially Professor Joseph Moran, who is the director of the Laboratory of Chemical Catalysis, who introduced me to this topic almost 6 years ago, because I'm a physical chemist by training and an engineer. So when I started to work on that, I had no idea what I'm getting into. But it has been very exciting, and I really appreciate that, and all the help of collaborators and funding, and you, of course, for your kind attention. And I will be happy to answer your questions.
Okay, Kamila, thank you for your interesting talk. We have a few questions on the chat. The first was from Veronica. She wants to ask you if gregorite and magnetite may be a byproduct of early metabolism.
I'm not really sure if they can be a byproduct. They can definitely be a promoter. These minerals are a product or of a process called of processes that are included in serpentinization. So these are geological processes that produce also hydrogen as a byproduct with water. But yeah, so I don't think they're direct products of primitive metabolism itself, but they're definitely a promoter or a catalyst, depending on your definition of catalysis.
Thank you. Rafał Szabla asked you, have you considered converting CO2 to the key substrates of your network by photoredox chemistry using the high prebiotic UV fluxes?
Yes, we have. We have not done that. This is, I would say, it's mostly due to some technical issues. Yes, it's not so. It's technically a bit fiddly, I would say, to have a reactor where you can use pressure and the UV light at the same time. So the option is there. We have not looked into that with probably high enough focus, but the option is there. I'm not saying no.
Okay, thanks. Tomek asked, could you provide the first metabolic particle?
I wouldn't say a particle. First metabolic molecule? Probably not, because even it's far too complex. Already water is a metabolic, it's part of metabolism. So, well, it depends where you draw the line. I wouldn't say you have a very sharp cutoff between the living and non-living. It's quite of a gray and a gradient sort of spectrum sort of transition. Formate is used by biology. Acetate is used by biology, and they are smaller molecules than pyruvate.
Okay, I understand this prebiotic chemistry is a really interesting topic. I want to ask, do you think it is possible to find any traces in fossils of this prebiotic chemistry, or it is totally not possible to find such traces?
That's a good question. I have no idea about fossils.
You know, to find, I don't know, some products or some I don't know, byproducts, molecules in the stones, rocks.
This is being done by the proportion of different carbon. I think it's carbon 12, carbon 12, carbon 13, or different isotopes. But from what I heard from people who work directly on that, this is not always so telling. So there are different variations between geological abiotic processes and biological processes. So basically, if you find a molecule that has an excess of some isotope of carbon, you could assume that this is of biological origin, but apparently not always. Again, this is something that I do not specialize in, and we would need some input from paleogeology specialists.
Okay, thank you very much for your interesting talk. This was our last talk in the session, and we go for the short, maybe not short, not very short break. It is 30 minutes, and we will meet at 6 p.m. On our keynote speaker, Jacek Krełowski from Astro. The talk will be in the topic of astrochemistry. First, it was planned that will be in Polish, but the talk will be in English, of course, and will be 45 minutes and 50 minutes for discussion. And chair will be Tomasz Zajkowski. And after this keynote speaker talk, we have the closing ceremony of the conference, and of course the presentation of the best poster. I invite you for these events, and now you have 30-minute break from the conference. Thank you very much. Thank you, Kamila, once again for your talk.
I thank you. Enjoy the break and see you later.
See you later. Bye-bye.
Bye.